All right, testing. I have a little talk about what BioProtocol is doing with AI agents, as well as giving a survey of the field of what research agents are doing in the more Web 2 land, without any crypto, without any DeSci, what agents are doing. So I'll get the slides up in a second. Here we are. My name is Sean Brennan. I joined Bio 5 months ago to lead the growing AI team at Bio. We have a team of 5 now, and we're building something called BioAgents. And what I'm declaring is that this is the start of agentic science summer. Like what is coming is many different teams in Web 2 are creating agents that are able to complete research tasks and discrete steps of knowledge work. And so I'll talk over these teams in a second, but I want to start with a quote by the head of OpenAI. He said, "We expect the impact of AGI to be uneven. Although some industry will change a little, scientific progress will likely be much faster than it is today. This impact will surpass everything else." So this is the head of OpenAI saying this, and I think they're looking at ways that AI agents are going to be able to be put in a loop and think about science, think about research tasks, and we'll talk about some of them today. I recently was lucky to attend an event in San Francisco put on by Compound VC, where over half these teams presented in person. And these are all what I call like Web 2 science, Web 2 AI research. So I'll talk about each one specifically in a second, but all of these teams are pushing the boundaries in each of their ways. They're state-of-the-art in agentic science. But again, they're not DeSci, they're not crypto native. And so what we're asking ourselves is, how does blockchain add to what they're doing, and how can we use what they're doing and accelerate it and scale it through token incentives, through on-chain knowledge graphs, through these different things that we're working on in crypto. First up is Google Co-Scientist. Here's just the flow of how they've set it up. It has multiple agents under the hood, and of all the ones here today, this is the only one that's closed source. And so they're basically spending a lot of money on servers for each. I don't know if anyone here has access to the early test version of Google Co-Scientist, but they're trying it out in alpha right now. And you can imagine for each chat window, they have tens, if not hundreds of servers working under the hood to answer this. And so this is all closed source. And what we're trying to do with BioAgents is replicate this, but also be open source and try to achieve the same results. So here's a little bit more on the timeframes. In the last panel and today, we talked about how biotech has very extended timeframes, and AI is a solution. Just the example here is that there was an idea posed in 2015 about a specific molecule in bacteria, and it took 10 years to have that experiment run. And just that same research question was posed to the AI scientist, and it basically created an experiment plan and was able to do this extremely fast. Another one to mention is Sakana AI Scientist version 2, which came out about 2 weeks ago. And this is a framework for running a research scientist as an agent. On these things, you can basically plug in any general model that you want. This could be O3, it could be Claude. And so this is a framework for running agents. And you basically put in a foundational model, and it has access to tools. And in this case, what they're doing is trying to create papers in the field of machine learning. And so what they did was start with an idea. They tune, I'll skip ahead to this next graph. So they start with an idea. It's like basically a tree search. And in each step, they have an AI LLM to look at the results of the previous step and see which of the trees to prune and which to continue down. Hyperparameters are just different variables in how an AI is built. And then the research agenda is actually, it's running code. And then ablation studies is something, ablation is a term for removing one specific piece from a study, or from like a LM's neural net, or like basically a piece of its knowledge, and then seeing how the AI performs without that one specific piece, and then trying out with the next piece. And on the whole, you get a view into what each specific piece of its knowledge base is contributing to the overall result. So this is really interesting to look at. OpenAI has something called Paperbench, where they're doing something very similar. They're trying to replicate known papers in the field of AI, basically saying, here's a a groundbreaking, noteworthy paper. Given its initial starting conditions, initial data set, can an AI replicate that paper? And they found that actually this wasn't using O3, it was using 4.0, and it was, in some cases, able to replicate it. Moving forward, Stanford has something around automating hypothesis. It's basically a loop where it's creating a hypothesis and a null hypothesis and thinking through this. And I have about 10 minutes total, so I'm going to breeze through this. I'm going to be writing like a Twitter thread with all these and links, so you'll be able to read that after if you're interested. Genentech has something called Prescient Design, which is an open source kind of lab within Genentech that put out, again, like an open source results of their experiments working with AI on antibody testing. And they basically had, this was one of the people that talked last week, and they had extremely successful results, and basically all of Genentech is going to start using this for their research. And the only inorganic side of this is something at Berkeley called A-Lab, stands for like automatic lab. For one of the things with AI is the discovery of molecules and discovery of the compounds is getting easier, but still the hard step is characterization, or actually creating that molecule, running tests on it, like the physical test. And so they work with, on the left here, I don't know if you can see, this is basically a pairwise reaction between different inorganic compounds, and they basically put these in a kiln or a furnace and create these, and it's entirely automated. So it's like a self-driving lab where there's like basically the grad students are only there to put in the initial ingredients, and then they let it run. And so they have a recipe, they have an iterative process, and it's pretty exciting. The two more here are Future House. It's super, like they're state of the art as far as creating an agent framework for automated testing of especially like benchmarks, like biology benchmarks, and grading agents against a known rubric. And it was cut off here, but Arc Institute has a model called EVO2, which is state of the art at genetic modeling and design. So, like going back to the Google co-scientist or the other ones, if they had access to this model, this foundational model, it changes the agent from being great at tool calling and conversation to being great at genetic design and basically a PhD level in this field. And the image here is just a few steps of the training process to create the EVO2 model. And lastly, yeah, these are the ones I went over today. And am I doing it on time? Switching over to what BioProtocol is doing, we're creating a fleet of BioAgents, and for all the ones before it, we're talking about one instance of an agent. Like one agent is able to do tasks at a certain cadence. It can run n tasks in a period of t time, and so if you give access to a lab to an agent like that, they're going to ask like, well, can I have two agents? Can I have the time that it takes to run a task, or create two results, and I get to choose the best result out of a selection. So we're pioneering agentic science over planetary scale compute and scaling these agents so that we increase the throughput of thought of each agent. And as a whole, we have a lot more compute time towards these problems. And so we are building a fleet of scientific task runners where there's going to be many, many agents, and they're all able to run tasks for the BioDAOs and for external parties to insert tasks. So instead of one agent able to think about a specific research problem, we have a fleet of 10,000 agents that are basically idle and open to accepting tasks. And the BioDAOs all have a lot of research and specific tasks to give the BioDAO, or to give the BioAgents. And the concept is very similar to AlphaFold, like folding at home did this. You can run a program on your computer, and it's just performing machine learning. It's just a mechanistic machine learning to solve protein folding. And Google DeepMind team and a company called Isomorphic Labs won the Nobel Prize in Chemistry last year for this. And we're doing something very similar, but instead of very, I guess, like formulaic machine learning, this is running agents at home. And we're inviting people to run agents that are part of the BioAgent fleet. And we can assign them tasks, and over time, we're scaling them on different companies like Prime Intellect, OLAS, and others. And we expect, because these are just Docker containers, they're very lightweight. Like, you don't need a full GPU to run these agents, and so we can scale to 10,000, hundreds of thousands, even millions of agents that are able to complete the research tasks that I was talking about before. And you can see, like, the reason that people would pay for this is that they get access to basically a research lab in college, or a biotech firm, or a startup, even a passionate individual. It doesn't have to run the DevOps. They don't have to create the agents. They don't have to set it up. And so just by paying in the bio token, they get access to insert tasks into the BioAgent fleet and get research done and just get the results at a very fast time frame. Here is just the discrete steps of the knowledge process. It just maps to the scientific method. People can start at one step and just run that single step, or they can run multiple steps. And like Kevin with Molecule was talking about, like IP NFTs, in theory, where you can start with a research paper, think about next steps, and go through the entire process of designing an experiment, running it, sending it to a cloud lab, and then if you have a positive result to your experiment, you can create an IP from that. Here's we're built on the ELIZA framework, and so this is what, kind of like under the hood, what the BioAgents are doing. They have different plugins. We have a hackathon going on right now, where it's still open to join. And if you want to create a plugin that gives a BioAgent access to a specific API, or maybe a new tool, it's actually very simple to add, so you can talk to me after if that's something you'd like to do. And to summarize, agentic science is a new field, and these new capabilities are leading to just an increase in technical thought. Like, the throughput of technical thought is increasing with AI agents. And using the open source frameworks that I talked about, especially like Sakana Scientist, there's a work from Stanford called Biomni, which is going to be open sourced in next month. I think it's going to be incredible. They talked about it last week and showed some demos, and it's basically we can leverage the open source community, like the Web2 open source community, and then we scale it with the bio token and our network here. And we're going to create a fleet of agents that are able to accelerate how fast experiments get done. And thank you.